Hi class, welcome back. So in this part of the series, we'll be talking about more on the principles of separation in capillary electrophoresis. Now remember, CE is known to us to have a very high efficiency and low consumption of reagents. It's very versatile, as you can see. We recap on the previous notes that it has different modes of CE, right? And the many approaches that's reported by the literature to optimize separation. Now keep in mind the important factors to achieve the best peak shape and separation here. Now we talk about the CZE, right? This is the first CE method that's invented and reported. And so as you know that, right, this capillary is a very fine small tubing here, and right, you have the different zones as a peak. Now recall that electrophoresis, right, is really, you can see an analogy as a swimming in electrical current. And the normal configuration is, right, you have this positive voltage here, and the positive ions will be pushed and they travel the fastest because of the opposite Coulombic repulsion, and the neutral ones are not affected by the voltage because it's not charged, and the negative ions are attracted backwards, that's why it's slowest. So we have something called the Vx, the electromigration velocity, right, and also the electroosmotic velocity, the electrophoretic velocity, right, that you need to take into account of. And the Vx here is really the sum of the VEO and VEP. So now what is the order of separation and elution, right? You can see it's cations and then neutral and anions. So the easiest way to optimize this electrophoregram is really to adjust the pH to change the charges on the analyte ions, right? Make sense? Now, we know that CZE is really easy to perform, and the separation is really based on different velocity, EP, and the analyte ions are separated based on the charge to size ratio, the charge density, and the charge can be adjusted by changing the pH, and the total migration velocity depends on both the VEO and VEP, and the migration time is given by Vx. Okay. Now VO, right, is really the velocity from the charges on the capillary wall, and the VEP is coming from the charges on the analyte ion. This is how you differentiate these two. So if your electroosmotic force, right, this flow, this EOF is more than the VEP, there's no need to have a pump because naturally, you just move along. Here you see two snapshots of the electrophoregram that Professor Lee, co-lecturer here, is going to talk about more in his research group by my seniors here under the J Chrome A. Now, normally anions and cations are analyzed separately, right? Because we don't want to mix them up, and they have a chance to also aggregate. But you can use the IC to detect anions and anions, right? IC meaning the ion chromatography. But you need to use different columns and different mobile phases. That's a bit frustrating sometimes. But CE allows your simultaneous detection of the anions and cations by using suitable separation medium. Of course, a buffer and everything else. So also the detection for the anions and cations, right? Although most of them, they do not absorb the UV ultraviolet light, it is still possible to detect by using the indirect UV detection. What does it mean here? Right, remember you have a window, right? A very small, narrow window at the long capillary, probably at 40 to 50 cm long. This is where you place a detector. Now, if you have a UV detector, what it does is, right, what is a direct detection? Direct detection means, right, something that fluoresces or get detected, and then nothing, and ding, and they pick it up, you see a peak. But indirect means, right, your whole capillary inside is really on the high first. And when your substance is there and it doesn't absorb UV, what happens is, right, there's a little drop. So when there's a drop, you know that something is there, right? You analyze. So more details can be found in the previous reference here. Okay, read further. Okay, next, the commonly used CE here is called the capillary gel electrophoresis, so we call it CGE. So these gels, right, they contain certain pore size, right? They are used to fill the whole capillary, okay? So interior is not hollow, it's this gel, and they're usually polymers, and depending on how they're cross-linked, the pore sizes can be controlled. Now, when the voltage, right, is applied, so these molecules migrate, right, through these pores of the gel, okay, and because they go through pores, it's a bit like a molecular sieving, you know, like you try to find your path and go push through the solid, and some of these molecules go through more easily, but others are blocked. Makes sense, right? Because you're trying to find the holes and tunnels and labyrinths. So because the gel are usually very viscous, I would say it's like a solid liquid interface, very viscous, it can also reduce convection. What it means is, right, less convection means you have less peak dispersion, right? Remember the heat dissipation? And so the gel will also improve separation by reducing the peak dispersion. And the gel also cover the surface of the capillary, right? So after you cover the surface of the capillary, it prevents the analyte molecules of getting adsorbed to the capillary surface, which means that it prevents adsorption, which means that it's just a natural flow of the analyte through the column. Okay, and by covering this with the capillary surface, right, it also help to eliminate any electroosmosis, right? So because the hydroxy group, remember on the siloxide, the SiOO group here, will not be able to dissociate to produce a double layer. So which means that in the CZE, right, this capillary gel capillary zone electrophoresis, you can reduce the analysis time by having a higher EOF here. Okay, but in this case, your capillary gel electrophoresis, your EOF can cause problems in two ways. Okay, what are they? The first problem is that it may affect the separation if you want to separate like purely, size-based separation, right? Another problem is that your EOF is large enough. It may destroy the gel, right? Because you see you have the EOF, the force that pushes through, and then the gel, is not rock solid, right? So the force is enough to break the gel, and once it's destroyed, you can't use it anymore. Okay, so to take note. Here, we want you to understand that using the appropriate gel is very important, right, to consider when you are taking CGE, the capillary gel electrophoresis, because the gel, first of all, need to be temperature stable. If it's not stable, there's no gel. If it melts, it doesn't form the whole medium. So when the voltage is applied, right, the heat is dissipated. And this gel should not degrade when a high voltage is applied. Just imagine when you have a high voltage means you have a lot of heat there. When you have a lot of heat, right, does the gel disintegrate? Think about it, or does it change chemically? The gel should also have some suitable size for separating the molecules, right, in the appropriate size range. Because like I said, if your sieve, right, your holes there are too tiny and your analyte is too big, just based on EOF, you will just crush. If the pores are too big, all the molecules go through without separation. If the pores are too small, all the molecules get stuck and might not go through it or take a very long time to go through. You don't want to waste time. Wasting time is like killing ourselves. Now, since the gel can also reduce convection and adsorption due to its high velocity, high viscosity, the molecules cannot diffuse easily, which is great. And with less diffusion, right, to broaden the peaks, right, the peaks can be very, very sharp, which is something we want. And so, therefore, it has a very high efficiency being achieved, even like 30 million plates per meter of capillary, very high plate number. So, the most commonly used gels in CGE are the polyacrylamide gels, and because they are widely used in the conventional slab gel electrophoresis. Now, the pore size, right, of these polyacrylamide gels can be quite easily controlled by changing the total concentration of the monomer. We call it the percentage T, and we come across this term quite a lot, percentage T, total concentration of the monomer, and the concentration of cross-linking agent, we call it percentage C. Okay, and by simply adjusting these two parameters, different pore size of the gels can be used to optimize the separation of molecules of different sizes. Okay, to understand. Now, these are just some structure of the polyacrylamide gel, and below are the structures of this the monomer, right, of the acrylic acid. You have the acrylamide put together, right. You have alternating monomer. You have the polyacrylamide PAM, okay, PAM, most commonly used in electrophoresis. Okay, next. So these polyacrylamide gels, right, in the CGE are always made using acrylamide as a monomer, and then your bis-acrylum as a cross-linker. Now, since both, right, of these two monomer have the amide group, the CN C double bond O NH group, right? The percentage T is calculated by adding up both the bis and acral and then divide by the total volume, right here. And because the percentage C, right, is calculated by dividing the bis by the sum of the bis and the acri and then multiply by 100%. Now, if the total concentration of the monomer is too high, what happens is many of these monomer molecules are packed together, which means that the pore size will be small, which means that the gel is more suitable to separate the small molecules. And if the total monomer concentration is low, there'll be more space, right? Makes sense. I don't have so much substance to make the gel, right? There'll be more pores. Then the pore size is bigger, which means the gel will be more suitable for separating larger molecules. Typically, about 5% C, okay, will give the smallest pore size for all the percentage T from gel electrophoresis. And since 5% C is most commonly used, okay, for initiation of the polymerization, different approaches can be used. What are the different approaches? Like in chemical initiations, right? Because, back to the basic concept of polymerization, we have got the chain polymerization, we have got the stepwise polymerization, right? Radical. You can read up more. I put the link up there for you. Now, if I want to initiate the polymerization, the suitable chemicals like ammonium persulfate ions or TMAD, TMED, right? They are commonly used there. Okay, a photochemical initiator. Now, in initiation here, the UV is commonly used. Okay. Yeah. So in the chemical peroxide here, what it does is it split the O single bonds very weak. You have an O dot oxygen radical, which is a good radical initiator. Okay, to start to attack the carbon-carbon pi bond of your monomer there. Now another approach here is not to use fixed gel, right? Which means you don't make the polymer inside the capillary. Now instead, what you do is you have a very dilute polymer solution, liquid that's used, and this polymer solution will have a different pore size based on the size of the molecule. So these polymer solutions, right, can also achieve size-saving separation. And although they tend to give a lower efficiency compared to fixed gels because of the high, diffusion and convection, right, flow, they are more convenient to use. And they can be pumped out of the capillary under pressure and replaced when necessary, right? Unlike the other one that's more solid there that reside inside the capillary. Okay, so the CG is the most common to separate large molecules, as you know. Proteins are large, so we like to separate proteins. DNA, all these are with CGE. Now, separating proteins, right? The different percentage T are used to prepare the size of different pore, right? So the 19:1 ratio for 5% T, right? The cross-linker, percentage C, right? It's commonly used. And for higher percentage T, the pore size will be smaller. So the gel is more suitable to separate small proteins. And for higher percentage T, the pore size is larger and the gels is more suitable for separating larger proteins, really large proteins. Okay. Okay. Another type of large molecules suitable for separations by CGE are the nucleic acids, right? Your DNA, RNA. So usually the DNA molecules are denatured by adding certain additives like urea, right? And then the molecules will have a long chain of bases rather than a double helix. So what you want is to separate the double helix in a single chain, single polymer, right? Being smaller. And the size differences will be based on the different base pair of the ACGT. And similar to the case of proteins, the 5% bis cross-linker is commonly used, which means a higher percentage T would mean that the gel molecules are packed more tightly, and hence the pore size is smaller for separating smaller DNA molecules. And the lower percentage T means the gel molecules are less tightly packed, the pore size are larger, and then the gel will be more suitable for separating larger DNA molecules. So it's no relative. Now this is another diagram here. We have a very simplified illustrations of the CGE separation of DNA. Okay, see strands of DNA. DNA molecules are nucleic acids, right? So they are negatively charged. And the polymer gels forms like a network, right, interwoven. And when the positive voltage is applied, the negative DNA molecules will be pulled, okay, by the positive voltage, holds them back. And small DNA can pass through the pores more easily, but the larger molecules will need to squeeze through the pores to find ways to make way through, and it takes longer time to pass through. So similar proteins can be denatured into peptides and amino acid, right? Because the bonds, you can cleave it. And so the gels can achieve the size-space separation of DNA and proteins. Here, the figures on top right shows a comparison of the slab gel electrophoresis with CGE. You see that the slab gel electrophoresis are typically done on a glass plate. Okay, so the gels are spread onto the glass plate, and then the analyte are dropped onto the gel, apply the voltage, and since the heat dissipation is not very efficient, the voltage applied is not as high in the CE because we want to make it too hot, right? It's not as high as the CE in capillaries. And hence the separation time is very, very long, typically like many hours. That means the analyte are then separated into bands, rather than peaks, because you can't separate them so easily. And hence it's also not accurately quantified. It can be done by integrating the areas under the peak, not so easy. Now, but the separation of the CGE, the capillary gel electrophoresis, is usually faster than the slab electrophoresis. It takes minutes, not hours. And the peaks in the CGE can be integrated easily to give an accurate quantitative result. You can measure how much of them we have inside, whereas in gel electrophoresis, the bands are not so easily to quantify. You can qualify but not quantify so accurately. And the figure below shows a log plot, okay, logarithmic plot of the molecular weight versus the normalized migration time. And you see that normalization is always done by dividing, right, the migration time by the reference migration time, right? So what is a reference migration time? We learn to talk about using a marker, remember, EOF marker, that's neutral. And this division, right, can be simply, the first peak. The first peak that comes out normally is the neutral molecule. So this plot is very useful if you have a separate proteins of different molecular weight. And then you can use the plot to determine the molecular weight of the unknown proteins using the normalized migration time. It's not that hard, but it's not too simple. Now, instead of using the fixed or cross-link gels, it is also possible to use polymer solutions and a size sieving separation. And this is sometimes called the non-gel sieving CE. Now, to use this method, there's some condition. It is not necessary to use coated capillary because there's no problem of destroying the cell, the gel, when high voltage is applied. And this example shows the separation of proteins, right, of molecular weight from 2,000 to 17,000. Very high, very heavy. And the polymer solution is only like 12% dextran. And this method is convenient and very fast. Okay, done by Professor Samley, properly. But the separation efficiency is not as high in the case of a fixed gel, since the diffusion and convection can be easier in solution. Now we know that the capillary gel electrophoresis was the main method used to sequence the human genome. We have to give credit to people, the inventor. And human genome is very complex, and to sequence all the DNA, a powerful separation technique is really required. So CGE can separate DNA with high efficiency much faster than the conventional gel electrophoresis. And by using multiple capillaries, we can do parallel separations. You can do it even faster. So these machines here run instruments uses a 96 capillary. In the year 2000, maybe before you're born, there was a rat race. Okay, let me drop the rat. There was a race, a very good competitive race to sequence the human genome. Okay, garnered high interest. So this person here, by the name of Craig Venter, had his own company. He was a very good entrepreneur, and he bought a few of these DNA sequencing machines. Then Francis Collins on the right here, this man worked for the National Institute of Health. You know, in the US, it's a very big research center. You call it NIH, gives out lots of big grant and funding. And he has an alliance with this institute in many parts of the world, in the country. Okay. Both of them first reported the draft of the human genome around the same time. So we can say that they are the first person who really have a scientific breakthrough here.